Yeah, my parents. That was it called again?
You came to this one?
It kind of is. Like, it's concerning if it's like there's no way coming back and forth.
Dude, I know.
Shall we resume our discussion? Okay, so we left off with a discussion of the oxidative phase of the pentose phosphate pathway, and we were going to lead into one of the uses of the NADPH that's generated through that oxidative phase. So NADPH is important for fatty acid synthesis that we'll talk about in a couple weeks, and cholesterol synthesis that we'll talk about after that. So NADPH is doing a lot in the cell. And this is just one important example of its function, is its role in maintaining antioxidant defenses through maintaining reduced glutathione. So glutathione is it's not a protein, but it's made of amino acids. Okay, so it's a tripeptide. There's a glycine, and a cysteine, and a glutamic acid. It's easiest to pick out the glycine because that doesn't have a side chain. So this is the nitrogen, this is the alpha carbon, and this is the carboxylic acid. So there's our glycine. Then we have N, this is the alpha carbon for the cysteine that has the methyl group and then the sulfur atom, and then the carboxylic acid. Then this is glutamate or glutamic acid, and you'll see that this is not a typical peptide bond, right? So here, this part is a peptide bond, NCC, NCC. The peptide backbone of the glutamic acid is here, N, C, and then this is the carboxylic acid. So it's the glutamate is bonded to the rest of the molecules through the side chain, not through the peptide backbone. Okay, so it's kind of a not super important, but I guess the thing to remember, there's not like a glutathione gene. Okay, this is made from amino acids, but it's not transcribed and translated into a protein like other proteins. Okay, all right. So the business end of the glutathione is this sulfur atom right here, and this sulfur atom, this can either be saturated with electrons, or in its reduced form as shown here, or it can be oxidized by making a disulfide bond with another molecule of glutathione. Okay, so that reaction is shown here. So you can take two molecules of the reduced glutathione, then in an oxidation reaction, these electrons can be stripped off to some other substrate, and then the sulfur atoms will then make a disulfide bond and be less saturated with electrons than they were over in this port. So this is a reversible reaction, and generally we want to have lots of the reduced form around because it's these oxidation reactions that are neutralizing hydrogen peroxide within our body. So let's see, and there's kind of a nomenclature, and I'll go back. So GSH, this is the reduced form, and that's referring to glutathione, and then the SH with the sulfur atom that's saturated with electrons. And then the oxidized form, this is often referred to as GSSG, referring to, so here's a glutathione and a sulfur, and then here's another glutathione and a sulfur. They're bound together. Okay, so that's where these terms come from. Okay, so if we have hydrogen peroxide generated, so we talked a little bit about where hydrogen peroxide can come from, right? So if you get single electron transfer from coenzyme Q to molecular oxygen, for example, you'll make superoxide. And then the mitochondrial, the manganese superoxide dismutase, that can make hydrogen peroxide from superoxide. Okay? So the hydrogen peroxide then, that can go on to make hydroxyl radical and make damage to the cell. Or if we have a robust pool of reduced glutathione around, that can be neutralized through the glutathione peroxidase reaction. So glutathione peroxidase, this is going to convert the hydrogen peroxide into water. While it's doing that, it's going to be oxidizing the glutathione to the oxidized form. Okay, another enzyme, glutathione reductase, then will regenerate the reduced glutathione. This then depends on reducing power of NADPH generated by the pentose phosphate pathway oxidative phase. Okay, so we also have the gluconolactone dehydrogenases would also be up here making NADPH. Okay, so this is just illustrating the importance of the pentose phosphate pathway in antioxidant defenses. This is my version of it, and then your Mark's version from your textbook is shown here. Are there questions about that? Okay. Okay, so let's switch over and talk about the regenerative phase. So the regenerative phase, that's taking the product of the oxidative phase, the sugar product of the oxidative phase, ribulose 5-phosphate. That can be converted into ribose 5-phosphate, and the ribose 5-phosphate can go on to be a substrate for phosphoribosyl pyrophosphate synthase and et cetera to make nucleotides. And we're going to talk about nucleotide synthesis in a week or so. So this will be elaborated on. So ribose 5-phosphate, this is kind of, if a cell is rapidly dividing and has a high nucleotide need, it's going to be doing this as a major function of the pentose phosphate pathway. So the ribulose 5-phosphate, this can be isomerized to the ribose 5-phosphate, or it can be epimerized, I think that's the right reaction, into a xylulose 5-phosphate. Xylulose 5-phosphate, this has an important role as a transcriptional regulator. So xylulose 5-phosphate is it's an intermediate of the regenerative phase, but it's also an indicator of high carbohydrates or carbohydrate excess, and it can remodel the cell to deal with those excess carbohydrates. Okay, so this we're going to talk about in a week or so. This I'm going to talk about in a few minutes. What I'll focus on for the next few slides here are the reactions that generate glycolytic intermediates from the 5-carbon sugars of the ribulose 5-phosphate. So there are two key enzymes, transketolase and transaldolase, and their names are might be a little familiar to you. So transketolase, that's going to be transferring a 2-carbon unit from one substrate to another. Aldolase, that's our enzyme that cuts a 6-carbon molecule into 2 3-carbon molecules. So transaldolase transfers 3-carbon bits of a molecule from a substrate to a product. So the basic goal is to turn 5-carbon sugars into either 3-carbon intermediate or a 6-carbon intermediate of glycolysis. Okay, and you can do that by adding and subtracting 2 carbons or 3 carbons from the different substrates. Okay, so I'm just going to show some examples. So the transketolase reaction, this is transferring 2 carbons from one substrate to another. So we've seen how we can get ribose 5-phosphate and xylulose 5-phosphate from ribulose 5-phosphate. All right. If we have these two substrates, the transketolase can take 2 carbons from one of them and add those 2 carbons to another one. Okay, so if you have 2 5-carbon molecules, you're making, I can't do this with my finger, but you're making a 3-carbon molecule and a 7-carbon molecule. Okay, so the 3-carbon molecule, glyceraldehyde 3-phosphate, that's familiar to us, right? That's an intermediate in glycolysis. This one can go down and be converted into pyruvates and what have you. Likewise, the same reaction, we could generate a 6-carbon intermediate of glycolysis if we had our xylulose 5-phosphate and then a 4-carbon sugar, erythrose 4-phosphate. If we take 2 carbons from the xylulose 5-phosphate and add them to the 4 carbons, then we get fructose 6-phosphate as a product. And not shown here, but we would also get the 3-carbon glyceraldehyde 3-phosphate when we take the 2 carbons away from the xylulose 5-phosphate. Okay? So I've got, did I have these arrows in your download here? Okay, if that's helpful. Okay, so that erythrose 4-phosphate, where could we get that? So we could get the erythrose 4-phosphate as a product of the transaldolase reaction. So for example, if we had the acetaheptulose 7-phosphate, the 7-carbon product of the transketolase reaction from the other page, we could transfer 3 carbons from the 7-carbon molecule, add it onto a glyceraldehyde 3-phosphate, then we would be getting a 6-carbon glycolytic intermediate. Okay, and we would generate erythrose 4-phosphate that could be substrate for a transketolase reaction. Okay, so, you know, it gets, well, it's very simple math, but some of the terminology of these are, you know, they're a little bit of a mouthful, but the concept is, I think, pretty straightforward. Okay, so if we wanted to make it complicated, we could try to make a balanced reaction. So if we wanted to, say, take glucose 6-phosphate and turn it into, go through the pentose phosphate metabolism and turn it all into glycolytic intermediates. And that would imply that we're not doing any of this, right? None of this ribose 5-phosphate is going out of the pathway to nucleotide synthesis. You could make a balanced reaction, and if you have 3 molecules of glucose 6-phosphate, you get 3 molecules of carbon dioxide and the different glycolytic intermediates, and all the carbons add up. Now, in reality, you're probably going to be diverting some of the carbon away from this, and so, you know, is a balanced reaction that useful? I don't know, probably not, but it can be done if you want to. Okay, are there questions on pentose phosphate pathway, regenerative phase? Okay, let's consider one of the functions of the xylulose 5-phosphate reaction, 5-phosphate. So the xylulose 5-phosphate, this can be substrates for the transketolase and enzymes and et cetera, and be converted into a glycolytic intermediate. But it has another role as a carbohydrate abundance sensor within cells. And basically, if you get a bunch of carbohydrate around, you get more through the pentose phosphate pathway, and you get more xylulose 5-phosphate buildup. So in situations of carbohydrate excess, adipocytes, so fat cells and cells of the liver, will convert that excess carbohydrate into fatty acids. Okay, and that process is kind of shown through in cartoon fashion here. We have to go through glycolysis to make pyruvate. Pyruvate goes through the pyruvate carboxylase reaction and then the pyruvate dehydrogenase reaction. The products of those reactions come together to make citrate. Citrate leaves the mitochondria. It's a substrate for citrate lyase to make acetyl-CoA, then acetyl-CoA carboxylase to make malonyl-CoA, and then malonyl-CoA is a substrate for fatty acid synthase. Okay, so let's not go through all that now because you're going to hear about that later. But basically, tissues can turn carbohydrates into fat. Okay. So where does xylulose 5-phosphate come in? Xylulose 5-phosphate comes in with not CREB, but CHREB. Okay, this, and I apologize for this, but so CREB, CREB, that's cyclic AMP response element binding protein. This is CHREBP, carbohydrate response element binding protein. And there's a little bit of overlap because they're both pKa substrates. They're both phosphorylated by protein kinase A, but they're regulating a different subset of genes. Okay, so the different domain structure is shown here. That's not terribly important. But the importance in metabolism is in their role, the carbohydrate response element binding proteins, in transactivating genes that are involved in lipogenesis. So we have the glucose entering a cell here. That's going to be converted. The more glucose there is, the more is going to be going into the pentose phosphate pathway, and the more of the xylulose 5-phosphate will be built up. Xylulose 5-phosphate, this acts as an allosteric activator of a different protein phosphatase than the insulin activated one. This is protein phosphatase 2. Protein phosphatase 2 erases the pKa phosphorylations on carbohydrate response element binding protein. That then allows it to go into the nucleus and transactivate genes that are important for lipogenesis. So this is a kind of a simpler view, and I added this in, just to note that high carbohydrates, that's also going to be a high insulin situation, right? So you've got the carbohydrate response element binding protein that enters into, one part of the promoter for these genes. Insulin receptor signaling, that acts in concert to increase their transcription as well. So some of this is familiar, phosphatidylinositol 3 kinase and AKT and protein phosphatase 1. And did you do translation and AKT control of translation through mTOR? This stuff? Okay, anyways, don't worry about that. Okay, so let's look at some of these genes that are upregulated in response to xylulose 5-phosphate. So pyruvate kinase, malic enzyme, citrate lyase, acetyl-CoA carboxylase, fatty acid synthase. So these are lipogenic genes, or genes that are involved in converting carbohydrate energy or carbohydrate carbon into a fatty acid. Okay, and the precise function of all of these genes, of course, we know what pyruvate kinase does, but the function of the rest of those genes will be covered in the lecture on fatty acid synthesis in a couple weeks. Okay, so bottom line, xylulose 5-phosphate, this is remodeling the transcriptional pattern of the cell to promote conversion of sugar into fat. Easy enough? Okay. All right, so why don't we do a few clicker slides, and then we'll talk about some disorders of fructose, galactose, and pentose phosphate metabolism. Okay, so ethanol metabolism has a couple of different options depending on how much you drink. Okay, so alcohol dehydrogenase and acetaldehyde dehydrogenase, this is a oxidative pathway of alcohol metabolism. So this is generating NADH. Both of these enzymes take a NAD plus and reduce it to NADH. CYP2E1, this is part of the microsomal ethanol oxidating system, or MIOS. And this is, it's a cytochrome P450 enzyme. Has Dr. Lotcher talked about those? Okay, so this is an enzyme. These enzymes have the kind of common feature that their expression is upregulated by their substrates. So when you drink a lot, you get more expression of the CYP2E1 and more of your ethanol gets metabolized through that pathway. So this is a reductive pathway, and this is actually, it's still making the same product, acetaldehyde, but it's using oxygen as an electron acceptor. So this is actually consuming NADPH reducing power. Okay, so alcohol dehydrogenase and acetaldehyde dehydrogenase, these are reducing NAD plus to NADH. CYP2E1 is oxidizing NADPH to NADP plus. Okay, all right. And these questions are pretty easy now that you've just, heard it a minute ago, but you got to kind of think forward to what this is going to look like on the final exam and if it's going to be so easy then. Okay, eliminate hydrogen peroxide. Is that, everybody agree on that? Okay. I'm not sure if we talked about the different isoforms of superoxide dismutase or not. But did we? Okay. Okay, so the manganese superoxide dismutase, that's the mitochondrial version, superoxide dismutase 2. There's also a cytosolic version and an extracellular version, SOD1 and SOD3, and they use copper and zinc as cofactors. So best answer here is manganese superoxide dismutase. Okay. This one, I'm not going to ask you about structures necessarily on an exam, but they're kind of useful to look at. Okay. Pink. This one. So there's nothing hanging off of the alpha carbon, so we know that's got to be glycine. Okay, so hopefully, yep, glyceraldehyde 3-phosphate, the best answer there. Okay, so best answer here is aldolase. And that's a good segue into our discussion of errors in fructose metabolism. So there are two inborn errors of fructose metabolism, and inborn errors of metabolism, that's referring to inherited loss of function mutations in enzymes that are important for that metabolic pathway. So with respect to fructose metabolism, there are only two that we have to worry about. a fructokinase deficiency, and then an aldolase B deficiency. Aldolase B deficiency. The fructokinase deficiency, this results in a condition called essential fructosuria. And essential fructosuria is benign. So it has no adverse symptoms whatsoever. Okay. You could get an abnormal laboratory finding of elevated fructose in the blood or elevated reducing sugars in the urine that may, you may think that the patient has diabetes if you're, so with urine dipstick sugar tests, some of them are specific for glucose and then others are just kind of looking generally for reducing sugars. So with essential fructosuria, you may get a false positive on a urine dipstick if it's just looking for reducing sugars because of the presence of elevated fructose. Now, with fructokinase deficiency, the fructose will accumulate in the blood and you'll have fructosuria, but that fructose has other metabolic fates. So remember that the fructose can be metabolized, albeit at a much slower rate, through hexokinase in muscle and other tissues. There we can generate fructose 6-phosphate, and this can be a substrate for PFK1 and go down through glycolysis. We can also urinate out excess fructose, this condition has no symptoms except for that potential false positive of the resulting from the fructose in the urine. So that is in contrast to hereditary fructose intolerance. Hereditary fructose intolerance is a deficiency of that liver-specific aldolase B enzyme. This has the clinical phenotype of having a hypoglycemia and tremulousness and seizures and kind of clammy skin and feeling unwell. And it will be preceded by ingestion of some food that has fructose. Okay, so people with hereditary fructose intolerance, this isn't something that, an infant who's only drinking milk, if they have this, it's not going to manifest while they're still on the milk, but it'll manifest when they wean and start eating regular foods. Okay, so the symptoms that result from the aldolase B deficiency, these are going to be dependent on ingesting fructose in the diet. So the mechanism for the aldolase B deficiency causing these symptoms is because the fructose 1-phosphate that's generated from the fructokinase reaction, that doesn't have any metabolic fate within the cell. Okay, so normally, if you think about phosphates within a cell, the P and the ATP here, metabolism requires those phosphates to go from one substrate to another at a pretty brisk pace, right? If you eat a bunch of fructose, it's going to be rapidly converted into fructose 1-phosphate, but this is then going to be stuck there. And the consequence is going to be the cell's entire phosphate pool is going to get trapped in fructose 1-phosphate. So, what's ATP synthase in the mitochondria going to use then? It's not going to have any phosphate around because it's all trapped in in this intermediate that doesn't have any metabolic fate. Okay, let's see. So there are some kind of general symptoms that would recapitulate some of the glycogen storage diseases, for example. One thing, it's referred to as a fruit aversion, or sometimes people have this. They eat fruit sugar, they get sick, and they avoid fruit from then on, or they have a strong dislike for it. So that's kind of one diagnostic clue, I guess, that you have the aldolase B deficiency. Okay, why would you get the hypoglycemia? Gluconeogenesis requires ATP, right? And if you don't have free phosphate around, you can't make ATP, so you're going to be hypoglycemic. Okay, so those are the two disorders of fructose metabolism. One is benign, one is not. In terms of galactose metabolism, we have lactase deficiency, which is a pretty common condition that results in diarrhea, flatulence after milk sugar consumption. So there's lactase deficiencies, and then there's the galactosemias. Lactase deficiency is a failure to break down lactose, so that doesn't result in galactosemia or elevated galactose in the blood. Right. Okay. So with the lactase deficiency, so in the gut epithelial cell, the lactase would normally be attached to that brush border with the enzyme activity projected into the lumen of the gut. If that activity is not there, the lactose then goes further down the digestive tract into the colon, and it's a substrate for bacterial fermentation. The bacterial fermentation then, this is going to make lactic acid. The lactic acid deionizes into lactate and a proton. Then you get water influx to balance out that proton concentration gradient. So you get a big osmotic effect and rushing in of water to balance out the proton imbalance. That big amount of water in the gut, that can cause the cramping and the diarrhea and etc. So how can these cells become lactase deficient? There's three main types, or three types. The most common is primary lactase deficiency, and this is a condition where you have normal lactase activity, but in adulthood you lose the activity. So you have milk tolerance as a kid, but then you lose it when you age. Secondary lactase deficiency, this is due to just anything that can damage these cells. So this could be a poisoning or a bacterial infection or something in the gut that's going to wipe out the function of those cells, and you get, a problem with digesting a lot of things, and lactase would be among them. Then congenital lactase deficiency, this is very rare, but this would be a inborn error of metabolism where you lack the lactase activity from birth onward. Okay, so lactase deficiency, this is, the treatment here is just to avoid milk sugars, and you can lead a pretty normal life. That's in contrast to the galactosemias. So there are galactosemias that involve loss of function mutations in galactokinase, the transferase, and the epimerase. So the least severe of them is galactokinase deficiency. The galactokinase deficiency, in the absence of the galactokinase, the galactose from a milk sugar meal, say, that galactose builds up, and the main consequence is that it enters into the eye and is a substrate for that polyol pathway that can generate, remember sorbitol was the intermediate sugar for glucose metabolism through the polyol pathway. You make galactitol with galactose. Anyways, the consequences are similar to too much glucose in the eye, where you get increased intraocular pressure and then increased glycation of proteins, and that can result in cataracts. So a patient with a galactokinase deficiency, they could, get cataracts, which you would normally see in a, 50, 60, 70-year-old. They could get cataracts in, 10 years old or 12 years old or something. Okay. The treatment here is to try to eliminate lactose from the diets, and it's a pretty good outcome if you can do that. Okay, so galactokinase deficiency, this is a non-classical galactosemia, and it's not life-threatening generally, and that's in contrast to the epimerase deficiency or the transferase deficiency. These result in a classical galactosemia. And these can be, quite severe and fatal if they're not recognized and an intervention isn't initiated. So the transferase deficiency, this was the first of the galactosemias to be identified, and this has symptoms of liver failure and hypoglycemia, and in some ways it's recapitulating the aldolase B deficiency. So these symptoms are resulting because the cell's entire phosphate pool is tied up as galactose 1-phosphate. Okay. Now the difference between this and the aldolase B deficiency is that this is going to be happening, from the infant's first meal, essentially, right? So it's happening when the infant doesn't have really choices about what it's eating. It's kind of locked into this diet. So that makes this. It's at a earlier developmental stage, and the infant doesn't have dietary flexibility like a older person would be. So this can be a quite severe situation if it's not identified and if there's not an intervention. The intervention is to eliminate galactose from the diet. And if that's done, there can be a pretty normal life and normal development. But if it's not done, if you have to spend a few months diagnosing it while the kid's still eating milk, then you can get some significant damage and it can be fatal. Okay, epimerase deficiency. This has essentially the same presentation as the transferase deficiency. However, the intervention and treatment is slightly different. With the epimerase deficiency, you don't eliminate galactose from the diet, but you have a galactose restricted diet. You have a tiny amount of galactose in the diet. Anybody have any thoughts as to why that is? Yeah?
Don't you still need UDP glucose for other cellular functions? So you don't want to, or not glucose, UDP galactose for other functions, so you don't want to completely eliminate it.
Yep, that's absolutely spot on. So you need UDP galactose to do those glycosylation reactions. And I have a couple of slides here that I think I gave you a slightly different version of this because I thought it was a little bit complicated, but okay, basically, so this is showing all of the different nucleotide conjugated sugars in the boxes. These are all used for glycosylation reactions, or substrates for glycosylation reactions. Don't memorize them all, except you'll notice there's not a nucleotide conjugated fructose anywhere there, so fructose isn't part of this. Okay, so we need this UDP galactose. If we have a deficiency in the transferase enzyme, we can still make UDP galactose through the UDP glucose and the epimerase reaction from dietary glucose, right? So here we can totally eliminate galactose from the diet. We can still make the UDP galactose that we need. If we have an epimerase deficiency, on the other hand, the only way that we can make UDP galactose is through dietary galactose. Okay, so in an epimerase deficiency, we need this, so the only way to get it is to try to fine-tune the dietary input of galactose so that you get just enough that you need, but you don't have this issue of tying up all the phosphates in the cell. Okay, is that understandable? Or any questions on that? Okay, so we'll conclude with the discussion of glucose 6-phosphate dehydrogenase deficiency. This is galactosemias are quite rare. Glucose 6-phosphate dehydrogenase deficiency, this is one of the more common inborn errors of metabolism. And as the name suggests, it's caused by loss of function mutations in the glucose 6-phosphate dehydrogenase enzyme, that first oxidative reaction in the oxidative phase of the pentose phosphate pathway. Now, the pathogenic mutations don't completely eliminate the enzyme activity, but they result in enzymes that are either unstable and have a shorter half-life, or enzymes that have a lower Vmax or a lower activity. Okay. All right, so the phenotype or the symptoms of glucose 6-phosphate dehydrogenase deficiency are going to be basically summarized as hemolytic anemia following an episode of oxidative stress. Okay, so these conditions can be clinically silent, and a patient will grow normally and into adulthood. But then if they're exposed to, say, an antibiotic whose metabolism generates a lot of oxidative stress, then they don't have the capacity in their red blood cells to maintain that pool of reduced glutathione, and the oxidative stress kills the red blood cells and causes lipid peroxidation and causes the cells to burst open. So you get an anemia, and it's hemolytic anemia. So there would be, ragged cells and bite cells and whatnot, not a macrocytic or a microcytic anemia. Okay, so just a few of those, like the sulfonamide drugs, antibacterial drugs, those are kind of classics, things that can precipitate that hemolytic anemia crisis in people with glucose 6-phosphate dehydrogenase deficiency. This condition is also sometimes called favism, and that's because fava beans, which are in the Italy and Libya and Mediterranean region, that's an important source of food, and the metabolism of the fava beans, I'm trying to remember the compounds in it. Anyways, there's sulfur-containing compounds in fava beans whose metabolism causes oxidative stress, and in people that lack this robust pentose phosphate activity, they can't generate enough NADPH to keep their glutathione in a reduced state to overcome the oxidative stress. So that's shown in a cartoon from Marx here. So we have the oxidative stress here generating the hydrogen peroxide. If the hydrogen peroxide is not neutralized, then it makes hydroxyl radical, and that can cause lipid peroxidation and cause the cell to rupture. So normally we have glutathione peroxidase is going to be neutralizing the hydrogen peroxide into water, but that glutathione peroxidase, that is depending on NADPH that's generated from the pentose phosphate pathway. Okay. This was a nice review article from a few years ago, and I don't know if you ever look at New England Journal articles, but there's a link if you do. I would encourage you to check them out. So some of them are, way deeper than you'll ever want to go until you're in your 5th year of residency or something. But a lot of articles are very, very accessible to first-year medical students, and this would be one of them. So. Okay. I have a few clicker slide questions, but I think I'll go forward and go through the clinical case in the last few minutes, and then if we have time, we can we can revisit the clicker slide questions. Okay. Does anybody have any impressions with this first presentation? This weight and the head circumference and everything, I guess I don't have the normals there, but there's these are below average, so suggesting a failure to put on mass and a failure to grow. Yeah.
Galactose phosphate uridyl transfer deficiency. Early onset got jaundice, and then hepatomegalia.
I like that. Yeah, so what would you, how would you confirm that hypothesis, or how would you test that hypothesis? Or anyone?
Do like, I don't know, you could do a biopsy, but I think you easily just switch to a formula that doesn't have galactose, and he gets better, then good to go.
That's yeah, that's definitely a good approach, and that's the cheapest thing to do. Just change the substrates and see if he improves. Right. Was there, or did you have some thoughts?
Yeah, like maybe like while being monitored, have them have lactose and then check the urine for galactose.
Ooh, yeah, give him a loading dose and then see what happens to it. Yep, I like that. Okay. Okay, so they did some labs, and the labs are showing essentially liver failure. And the bilirubin, that would account for the jaundice, right? So AST, ALTs are up. Now, total serum protein and the serum, these, again, that's reflecting liver damage because most of the proteins in your blood are synthesized by the liver. And that would include the coagulation factors, so the coagulation is, well, it's barely normal. Hypoglycemia, not anemic and no infection. And the urine is positive for reducing sugars. Okay. So, is the patient diabetic? Okay. He's not going to be peeing out glucose with this level of blood glucose, right? You have to have you have to be in the hyperglycemic area before you'll start peeing out glucose. Okay, so what should we do with this patient? Pull some of his enzymes out and check their activity. So given those laboratory values, what is the most likely cause of the child's symptoms? Transferase deficiency or epimerase deficiency? Epimerase deficiency. Okay, good. What would you recommend then? Galactose restricted diet? Everybody agree? Okay, good. Yeah, so this is a case where, so that it was diagnosed as epimerase deficiency. It was identified early in the child's life. There was this, I was going to say a relatively simple dietary intervention, but there's going to be a little fine tuning there with getting just the right amount of galactose. But it's a pretty cheap intervention, and it made a very significant improvement in the quality of life. So the child is now, growing where the kid wasn't growing before, and doesn't have these liver failure. Okay, questions about that one? Okay. Let's talk about this for just a moment. So glucose 6-phosphate dehydrogenase, that enzyme is expressed in every cell in our body. Right? But people with glucose 6-phosphate dehydrogenase deficiency, the deficiency manifests itself only in red blood cells. So you get, when you get oxidative stress, you get hemolytic anemia rapidly. So why do you think that is? Yeah.
The iron in red blood cells kind of predispose these red blood cells to oxidative stress, and so if they're not reduced, then they would be more susceptible to oxidative stress that wouldn't be able to be addressed by NADPH produced through the pentose phosphate pathway.
I think that's a good thought, and that's definitely part of it, is that the red blood cells have oxygen, and they also have iron, so they're a big source of oxidative stress. But there's kind of a deeper level when you think about like how could any other cell respond, and how could a red blood cell respond to oxidative stress? Did you have a?
Well, this is a stab in the dark, but is it just because like red blood cells are like limited to their energy source being glucose?
It has to do with specific organelles that are lacking in a red blood cell, right? Red blood cells don't have a nucleus. So if you have a liver cell, say, that has decreased activity of glucose 6-phosphate dehydrogenase, and that liver cell encounters oxidative stress, it has the capacity to increase expression of that enzyme. Or if the enzyme has a shorter half-life, then it can just ramp up the transcription and translation of it. With a red blood cell, once that red blood cell leaves the bone marrow and poops out its nucleus, it doesn't have any capacity to resynthesize new glucose 6-phosphate dehydrogenase. Okay, so it's just stuck with what it's got. Does that make sense? Okay. Okay, sorry for going over a little bit, but I thought that was a useful discussion. Okay, please come see me or shoot me an email if you have questions about any of this material. Same kind of.
It's just in here, right?
Well, I guess it is, yeah.
Like this table's downstairs. Not the like, what's the prestige?
I think Skildum should
I need to go get my lunch.
Then there's other ways to get this, then it was, in my mind, I'm thinking it's the same issue where you're not gonna get glucose because this reaction can't go forward.
But you can get UDP glucose from glucose 1-fridases. Like, remember how we synthesize glycogen from glucose?
Right.
So that's where you can get UDP glucose. That
What's going on? Something good?
Okay.
So,
Oh, I see, because it's like a back door is coming into here.
yeah,
I
so
see.
yeah,
Yep.
you would have galactose in the blood, and any UDP galactose would come from this.
Okay.
Make sense?
That makes sense, yeah, thanks.
I work fast now.
Dude, so do I.
Like when I first couple weeks when I studied with other people, it's like you get distracted, and you
Same.
goof off kind of.
Yeah.
And at home, when I'm at home, like